Hey guys, Dan and Manik here, and we're going to talk about mitosis and the cell cycle. Mitosis is a form of asexual reproduction. During mitosis, a cell divides into two genetically identical cells. Cells need to divide in order for an organism to grow, repair itself, and in some cases, reproduce. Here we will show you the cell cycle, as well as all four main phases of mitosis. Here you can see the life cycle of a cell. Most of the cell's life is spent in interphase. There are three parts of interphase: G1, S, and G2. During G1, the cell spends its time growing and carrying out its functions. During the S phase, the cell's DNA is replicated. Next, the cell goes into G2, where the cell continues growing until it reaches its maximum size. Certain cells do not replicate regularly. These cells are in the G0 phase, where the cell carries out bodily functions and other tasks needed to help the organism. At this point, the DNA is in a loose state called chromatin, and the cell starts dividing in a process called mitosis. Mitosis is made of four main phases. The first is prophase. During prophase, the chromatin condenses into a dense state called chromosomes, and the nuclear membrane and nucleolus disintegrate. Next, the centrosomes move to different sides of the cell, and spindle fibers form from them. The spindle fibers then attach to chromosomes at the centromere. The next phase is called metaphase. During this phase, the spindle fibers push and pull the chromosomes till they align at the center of the cell. Next, during anaphase, the spindle fibers shorten, pulling the sister chromatins apart so that half the chromosomes are on each side of the cell. Finally, during telophase and cytokinesis, the two chromosomes are pulled to opposite poles, and the nuclear membrane and nucleolus reappear. Then the DNA relaxes back into chromatin. Then, a ring of protein fibers pinch the cell into two new daughter cells. The result is two genetically equal daughter cells. These cells are now entering G1 and will repeat the cell cycle. Throughout the cell cycle, there are three checkpoints to regulate the activity of cells. If a cell does not meet the requirement of one of these checkpoints, the cell will either fix the problem or eliminate itself. The three checkpoints are between G1 and S, G2 and mitosis, and between metaphase and anaphase. The G1 to S checkpoint is the most important and is called the R point. At this checkpoint, the cell checks if it still has its DNA intact. This makes sure the cell does not replicate bad DNA. Another checkpoint is between G2 and mitosis. During this one, the cell checks if the DNA has been replicated before beginning mitosis. The other checkpoint is between anaphase and metaphase. Here the cell checks to make sure the DNA is aligned properly at the equator. This is important to make sure the cell divides equally, and each new cell has all 46 chromosomes. These checkpoints are controlled by CDK proteins. When a growth factor binds to a receptor protein, it activates CDK synthesis. Then the CDK is activated by cyclin, and it phosphorylates the protein RB so that DNA replication can then occur. When DNA is damaged, the p53 protein blocks the phosphorylation of RB so that the DNA cannot be replicated. Sometimes the receptor protein mutates so that cyclin keeps being synthesized. This causes DNA to be repeatedly duplicated and can lead to cancer. The same effect occurs if the cyclin is mutated and therefore is always being synthesized. Other times, the RB is mutated so it does not need to be phosphorylated in order for the DNA to be replicated. This leads to the replication of bad DNA and sometimes cancer.